Hello everyone, my name is Niyati Seth and thanks for watching Edupedia Word videos. My topic for presentation is the cell. I have divided this chapter in 3 sections. So let's proceed towards the first section, that is the cell. Cell is the unit of life. Cell is a fundamental unit. Cell is a basic structural and functional unit of life. When we look around, we see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that our inanimate things does not have, which a living thing has? The answer to this is the presence of basic unit of life, the cell, in all living organisms. All living organisms are composed of cells. Some are made up of a single cell known as unicellular cells, and while other, like us, composed of many cells called as multicellular organisms. Now question arises, what is a cell? Unicellular organisms are capable of, first, independent existence, and second, performing essential function of life. Always note one thing, that anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structure and functional unit of all living organisms. Now come to organization in multicellular organisms. As we know, our body as a whole in one organism. However, many parts make up that whole. As you consider the various levels of body, you understand that a large number of parts are within parts. It's akin to looking at a pine tree. At first, you notice entire tree as a whole organism. However, as you look closer, you notice the branches. Looking at the twigs on the branches, you notice each needle on the twigs. The same analogy holds for the human body. First, you notice the entire body. Next, you see that entire body is made up of parts and organs, and each of those organs is made up of variety of tissues. So, many cells comprises to form tissue, and many tissue or variety of tissue comprises to form organs, and many organ comprises to form organ system, and many organ system or variety of many organ systems comprises to form organisms. This is a typical diagram of animal cell. As you can see, this is a cell membrane. This part is cell membrane. Cell membrane holds and protects the cell, see, and it controls the movement of material into and out of the cell. That's why its nickname is gate of the cell, and it is generally a double layer of phospholipids. So it is made up of phospholipids. Then comes cytosol. Cytosol is a soup within which all other cell organelles reside, and where most of cell metabolism occurs. Cytosol is full of protein that controls cell metabolism, including cell signal transduction, glycolysis, etc. And then comes cytoplasm. Cytoplasm is a watery or gel-like material in which cell parts move or float. That's why its nickname is area of movement. Then comes mitochondria. Mitochondria provides energy to cell for movement, for division of cell, and mitochondria generate chemical energy similar to type of energy you get from battery. That is why it is also known as powerhouse of the cell. Then comes chloroplast. Chloroplast contains chlorophyll, which is a green in nature, and it captures the energy of sunlight and uses it to produce food for the cell. And that's why its nickname is food producers for the cell. And then comes vacuole. Vacuole is a storage for food and water and chemical. That is why it is known as storage tanks. And then comes nucleus. Nucleus is a head of a cell, or it acts as a brain of the cell, and it regulates and controls all cell activities. That is why it is also known as control center. And then comes the nuclear membrane. This is the membrane that covers the or surrounds the nucleus. It protects the nucleus and it controls the movement of material into and out of the nucleus, because it is a brain of the cell, and that is why it is also known as gate of the nucleus. And then comes chromosomes. Chromosome, they are present inside the nucleus, and it directs cell activities, and that is why it is also known as director of the cell. Discovery of cell. Robert Hooke was the first one who discovered the cell. Hooke discovered a multitude of tiny pores that he named cells. He saw little compartments and he called them cells. Now come to cell theory. Cell theory was formulated by M.J. Scleddon and Theodore Swan. Before 17th century, no one knew that cell existed. Cells were not discovered until after invention of microscope in early 17th century. One of the first microscope was made by Dutch drapery store owner Anton van Leeuwenhoek. With his handheld microscope, Leeuwenhoek became first person to observe and describe microscopic organism. In 1665, English scientist Robert Hooke used a microscope to examine thin slice of cork and described it as a cork little boxes. Little boxes reminded him of small rooms in which monks used to live, so he called them cells. In 1838, German botanist Scleddon, this person, he studied variety of plants and concluded that all plants are composed of cells. Next year, a German zoologist, Swan, he reported that animal cells are also composed of cells, and he said that it is a cellular basis for all life. Virchow, he induced that animal arises only from an animal cell and plant arises only from plant cell. Cell theory, it is a combined work of Scleddon, Swan and Virchow make up what is now known as modern cell theory. The main postulates of cell theory are: all living organisms are composed of cells and products of cell. Second, all cells arise from pre-existing cells through the process of cell division. And third, the body of living organism is made up of one or more cells. Now come to shape of cells. There are variety of shape of cells. One is chlamydomonas, cartilage cells, bacteria, epithelial cells, striated muscle fiber, leukocyte, smooth muscle fiber, diatom, fat cell, human sperm, smooth muscle cell, nerve cell and bone cell. So come to smooth muscle cell. A smooth muscle cell, each fiber cell can contract independently and is virtually innervated by a single nerve ending. These fibers are insulated from each other by a covering of collagen and glycoprotein fibrillae. Seldom exhibit spontaneous contraction. Examples, you can say ciliary muscle of eye, ancillary muscle of iris and piloerector muscles. Second shape of cell is ovum. Ovum, in appearance and structure, the ovum differs little from an ordinary cell. Despite its large size, it's only animal cell you can see with the naked eye. It is generally spherical, non-motile gamete with yolky cytoplasm. Human ovum is surrounded by a number of egg envelopes. This is a vitellian membrane. It is an inner and thin and at the same time it's transparent and it is secreted by itself. And this is zona pellucida. This is zona pellucida. It's a middle, thick, transparent and non-cellular. It is secreted partly by follicular cells and partly by oocytes. And this is corona radiata. This is a corona radiata, and these are the follicle cells of corona radiata. It is a very thicker coat, and this is the perivitelline space. Between vitelline membrane and zona pellucida there is a narrow perivitelline space. And human ovum is a microlecycle with large amount of cytoplasm. Sperm. The term sperm refers to male reproductive cells. Those primary function is to fuse with ovum, to restore diploid and to transmit paternal characters to the offsprings. It is a haploid male gamete whose primary function is to fuse with ovum, and this is a typical structure of sperm. Mammalian sperm is minute and cannot be visible by naked eyes. It is microscopic, flagellated. As you can see, it has a flagella, which is also known as tail, and it is a motile gamete because it has a tail, so it has the ability for movement, and basically it is formed of 4 parts and each perform a specific function. This is the head part, this is the mid piece, and this is the tail part. Head. Generally it is flat and oval. Head is formed of 2 parts. One is acrosome. This is acrosome and this is nucleus. Acrosome, just break the terminology and you will get to know the meaning. Acrome is extremity and soma means body. And soma means body. It is a cap-like pointed structure present at the tip of the nucleus. During a sperm entry, the acrosome secretes a lytic enzyme called hyaluronidase, which help in penetration of ovum. Then comes the second part of the head, that is nucleus. Nucleus is generally long and narrow but is flat in mammals. Then comes the middle piece. Middle piece lies just behind the neck and is cylindrical in nature. It contains many mitochondria. That's why middle piece of sperm is also known as powerhouse of the sperm. And then comes the tail part. This is the longest part of sperm. It's slender and tapering. Then comes epithelial cell. Epithelial cell is one of 4 basic types of animal tissue, along with connective tissue, muscle cell, nerve cell, etc. Function includes secretion, selective absorption, protection, detection of sensation. In general, simple epithelial tissues are classified by the shape of the cell: cuboidal, columnar, squamous and stratified squamous. Simple cuboidal, these cells may have secretory, absorptive, excretory functions. For example, include small collecting ducts of kidney and pancreas. Then comes simple columnar, found in areas with extremely high secretive or absorptive areas. They possess cellular extensions like microvilli in small intestine. And then comes simple squamous. It is found in living areas where passive diffusion occurs. For example, walls of capillaries. And now comes stratified squamous. These are simple columnar epithelial cells whose nuclei appear at different heights, giving the misleading impression that epithelium is stratified, and that is why they are also known as pseudostratified epithelium cells. Now come to nerve cells. Nerve cells are found in nervous system. These are specialized cells designed to stimulate other cells in the body in order to communicate. Neurons are excitable, which means they function by using electrical stimulation. Through this electrical message known as action potential. Now let's see its structure. These are dendrites. These are dendrites. A neuron may have up to 200 to 300 of these short branching projections. And then comes axon. This is the axon part. Nerve fiber may link to cell bodies or dendrites of other neurons. Then comes synapses. Synapses are the gap that separates a fiber end bulb from an adjacent cell body or a dendrite. This is a myelin sheet. This is the myelin sheet which covers the axon. It is a fatty coating which speeds up the nerve impulse transmission. And this is the node of Ranvier. These are those gaps in the myelin sheet which help in the conduction of nerve impulse. Now come to fat cells. They are also known as adipose cells or adipocytes or lipocytes. It is a loose fibrous connective tissue. An adipocyte is an animal tissue or cell whose particular function is to storage of triglyceride. As you can see, that triglyceride is stored in a large central area. This yellow part is triglyceride. The central area of the cytoplasm of adipocyte, their nuclei that is why their nuclei are peripherally located. Food that is excess to requirement is converted into fat and is stored within adipose tissue. Adipocytes also has a protective function as it provides mechanical protection and support around some organ. For example, kidneys. Then comes RBCs. RBC is red blood cells. They are round with a flattish indented center. They are like you can say donuts without a hole. They carry oxygen. Red cells contain hemoglobin which permits them to transport oxygen. Hemoglobin, aside from being a transport molecule, is a pigment. It gives the cell their red color. Additionally, erythrocytes are enucleated. That means they don't have nucleus. This extra room allows for more hemoglobin to be stored in our RBCs. Now come to Spirogyra. Spirogyra is a genus of filamentous green algae of order Zygonometales named for the helical or spiral arrangement of the chloroplast that is diagnostic of the genus. These are freely flowing filamentous algae and are characterized by ribbon-shaped chloroplasts that are arranged in a helical manner. That's why a name is derived from a spiral arrangement of chloroplasts in these algae. See, this is the centrally located nucleus. This is the middle layer. This is the middle layer which is made up of pectin. Then comes the cell wall. This is the cell wall. Dark green color is a cell wall. It is the inner layer of cellulose and outer layer of pectin, which is continually being replaced. Then comes pyrenoid. These are the pyrenoids. These dark spots on the ribbon shape are pyrenoids. Pyrenoids are concerned with starch formation, and these are the vacuoles. These are the vacuoles that contains a solution of sugar, mineral ions and amino acids. And these are the cytoplasmic strands. These are the cytoplasmic strands that holds the nucleus in a central position. Then comes the amoeba. Amoeba have a single cell that looks like not much more than cytoplasm held together by a flexible cell membrane. Amoeba is one of the most enigmatic life forms. They are legendary as the ultimate shape shifters. They have a cell membrane. This is the cell membrane. It is a very thin layer of protein and fat that surrounds the amoeba. It allows some substance to pass into the cell and blocks other substances which are not required for the amoeba. And this is a nucleus, brain of the amoeba, major organelle of the amoeba located centrally. It controls reproduction and many other important functions. These are the pseudopods. You can say that they are temporary feet of amoeba, and amoeba uses to move around and to engulf food. Then this is the cytoplasm. It is a jelly-like material that fills most of the cell. This is the food vacuole. It is a cavity within the amoeba in which food is digested. This is a contractile vacuole, a cavity within the amoeba that excretes excess water and waste. The waste is brought to this contractile vacuole and then eliminated out from the amoeba. Now come to bacteria. Bacteria, all cells have three main components. First is DNA, that is nucleoid, and which has genetic instructions, and this is the surrounding membrane. This is the surrounding membrane, or also known as cytoplasmic membrane, that limits access to the cell's interior, and the fluid inside is a cytoplasm, that is between DNA and membrane, where all metabolic reactions occur, and especially protein synthesis, which occurs on the ribosomes. Bacteria also often have these features, like cell wall. Cell wall resists osmotic pressure, and cell wall is composed of peptidoglycan in bacteria, and they are of two types of bacteria. One is gram positive and other is gram negative. They both are defined by the structure of cell wall. There is a dye named gram strain. When gram strain binds to the peptidoglycan, it can be seen under microscope. This is the gram positive and gram negative bacteria. Gram positive, this is a typical structure of gram positive. Many layers of peptidoglycan, as you can see, they have many layers of peptidoglycan which is anchored to cell membrane by teichoic acid, and in gram negative bacteria, it has 1 to 2 layers of thin peptidoglycan layer. And they also have periplasmic space. This is periplasmic space. It is a space between cell membrane and cell wall. It contains enzymes and other proteins such as chemoreceptors for sensing environment, and outside peptidoglycan layer is the outer membrane. This, as you can see, this is the outer membrane which is made up of lipopolysaccharide and protein. It is pierced with the porins also, or which are also known as protein channels. This is a typical diagram of gram positive and gram negative. See, this is a gram positive bacteria. It has many layers of peptidoglycan which is anchored to cell membrane by teichoic acid. This is a teichoic acid. This is the distinguishing feature of gram positive bacteria. And now come to gram negative bacteria. It has, as I said, that it has 1 to 2 layers of thin peptidoglycan. It also has a periplasmic space. This space is periplasmic space, which is a space between cell membrane and cell wall. It contains enzymes and other proteins such as chemoreceptors for sensing environment. And outside the peptidoglycan layer is the outer membrane which is pierced with porins. See, as you can see, these are the porins or the protein channels. And brown lipoprotein and porins are the distinguishing feature of gram negative bacteria. Now come to plant cell. Plant cells are eukaryotic cell, that is the DNA in a plant cell is enclosed within the nucleus, and the most important distinctive structure of plant cell is the presence of the cell wall outside the cell membrane, which provides support and rigidity to the plant cell. And the cell wall mostly constitutes of cellulose, which provides support and rigidity. Plant cell also contain many membrane bounded cellular structures. And there are wide range of operations that its cell organelles operate, like producing hormones, enzymes, and all other type of metabolic activities in the cell. This is a typical diagram of plant cell. As you can see, this is the cell wall. This dark green color is the cell wall. As you can see, this is the cell wall. This is the outermost rigid covering of the plant cell. It is a silent feature of a plant cell because animal cell does not possess cell wall. So it is a distinguishing feature of a plant cell. Then just behind the cell wall, cell membrane is there. Cell membrane is also known as plasma membrane. It lies just behind the cell inside the cell wall. Even animal cell also has cell membrane. And then comes cytosol or cytoplasm. This is cytoplasm, which is jelly-like and which constitute all other cell organelle. And this is nucleus. This is nucleus, or also known as brain of the center. This is the control center of the cell. It's a membrane bounded structure which contains hereditary material, that is DNA. And this is mitochondria. Mitochondria are powerhouse of the cell, which provides energy to the cell for carrying out respiration, division, and reproduction. These are the vacuoles. This is a vacuole. Vacuole is a temporary storage center of the cell. Golgi, this is the Golgi body. This is the Golgi body, channel like it is a Golgi body. It is a unit where proteins are sorted and packed. And these are the ribosomes, where it is a structure that assemble proteins. And this is ER or endoplasmic reticulum. This is smooth ER and this is rough ER. Rough ER which has ribosomes on it, and smooth which does not have ribosomes on it. And it is a large network of interconnecting membrane tunnels. And this is a chloroplast. Chloroplast is an elongated or disc-shaped organelle containing chlorophyll, which is a green pigment. And the process of photosynthesis occurs in the region of plant cell. Now come to animal cell. These cells tend to be larger than the cells of bacteria and have developed a specialized packaging and transport mechanism that may be necessary to support their larger size. This is the anatomy of animal cell. As you can see, this is a cell membrane, this is nucleus, nucleolus, this is vacuole, this is Golgi complex, this is endoplasmic reticulum, this is mitochondria, and this is lysosome. Just similar to plant cell, except it does not have cell wall. So plant cell and animal cell. Plant and animal cells are composed of same fundamental constituents. For example, nucleic acids, protein, carbohydrates, lipids, and various inorganic substances, and are organized in the same fundamental manner. A characteristic of their organization is the presence of unit membranes, which are composed of phospholipids and associated proteins. Both have a nucleus and cytoplasm, plasma membrane, Golgi apparatus, mitochondria, endoplasmic reticulum, and ribosomes. But where differences lies in the shape, cell wall, vacuole, chloroplast, and food storage and centriole. Shape, first consider shape. Animal cells, they don't have fixed shape because they don't have cell wall to provide support or rigidity to the cell, whereas plant cell have fixed shape and cell wall. They don't have cell wall because and that's why they don't have fixed shape, and on the contrary to this, plant cell have cell wall. Vacuole. they do not have vacuoles. Animal cell, they do not have vacuole, or they may have, but a very small vacuole. Whereas in plant cell, they have very large vacuole. Now come to chloroplast. Chloroplast, they don't have chloroplast, animal cell, because they cannot synthesize their own food. They are dependent on the plant cell, and so on the contrary to this, plant cell, they have chloroplast. Now what is food storage? In animal cell, food storage is glycogen, and in plant cell, food storage is starch. And centriole, they have centrioles which help in the cell division, and on the contrary to this, plant cell, they don't have centrioles. This is the differences between animal cell and plant cell. As we have read that cell wall is absent in animal cell, that's why they don't have a fixed structure. And cellulose in any form is also absent, because cellulose is a main component of a cell wall. And on the contrary to this, plant cell, cellulose is a main constituent of a cell wall. And second is cytoplasm, which is present in animal cell, is very denser and more granular, and it occupies most of the cell in the space in the cell. And in plant cell, cytoplasm is pushed to the periphery and forms a thin lining against the cell wall. Third, plasts are absent, or chloroplasts are absent in animal cell, whereas in plant cell, they are generally present. Vacuoles are absent, or if they are present, then they are very small and temporary, and it is only concerned with excretion or secretion. Whereas plant cell, vacuoles are large and prominent, maybe one or more. And centrosome is present with one or two centrioles. Centrioles are basically meant for cell division, and whereas in plant cells, centrosome is absent, but two small clear areas called polar caps are present. These polar caps participate in cell division because they don't have centrosome. So in case of instead of centrosome, they have polar cap, which help in cell division. They have Golgi bodies, very prominent and very complex, near and that is present near the nucleus. Whereas several subunits of Golgi apparatus called dictyosomes are present in plant cell. They reserve food stored in the form of glycogen, and plant cell, they reserve their food stored in the form of starch. So now come to prokaryotes and eukaryotes. This is a typical diagram of prokaryote and eukaryote. As you can see that in prokaryote, they have cell wall which is made up of peptidoglycan, and they have circular and supercoiled nucleus. They have plasmids, they have cytoplasm which are rich in ribosomes, and they have cell extension also, such as flagellum. And eukaryote, this is a eukaryote typical structure. They have cell membrane, nucleus, nuclear membrane, cytoplasm, SER, that is a smooth endoplasmic reticulum, RER, that is rough endoplasmic reticulum, Golgi body, lysosomes, and mitochondria, which is the site for cellular respiration. Now come to prokaryotes. Prokaryotes, they have cell extensions. As you can see, these are the cell extensions, and such as cilia and flagellum. They have cell membrane, cell wall, and capsule, ribosome, and cytoplasm. Eukaryotic cell structure, this is a typical diagram of eukaryotic animal cell. As you can see that they have cell membrane or plasma membrane, mitochondria, peroxisomes, cytoskeleton, free ribosomes that are floating on the cytoplasm, centrioles, lysosomes, Golgi apparatus, nucleus, and secretory vesicles. This is the similarity and dissimilarity between eukaryote and prokaryote. Eukaryotes are 10 to 100 micrometer in diameter, hence they are bigger in size. And on the contrary to this, prokaryotes have just 2 to 2.0 micrometer in diameter, hence they are very smaller than eukaryotes. Eukaryotes have membrane bounded nucleus and nucleolus, whereas eukaryotes, in prokaryotes they have unbounded nucleoid, means a circular DNA just float inside the cytoplasm. They have complex appendages, eukaryotes, and whereas prokaryote, they have very simple appendages. And in eukaryotes they have linear DNA with histone proteins in it, and whereas prokaryotes, they have circular DNA. In eukaryotes, all cell organelles like Golgi apparatus, endoplasmic reticulum, vacuole, nucleus, they all are membrane bounded, whereas in prokaryotes they are not bounded by membrane. And in eukaryotes they have membrane receptor for sensing the environment, whereas prokaryotes, they don't have this feature. And eukaryotes, they divide by mitosis and meiosis too, and prokaryotes they divide by simple binary fission. And cell wall is very simple in eukaryotes, whereas in prokaryotes it's a very complex cell wall. And in eukaryotes they have very big ribosomes, whereas in prokaryotes they have very small ribosomes. And cytoskeleton is a distinguishing feature of eukaryotes, whereas prokaryotes, they don't have cytoskeleton. So this comes to an end, and we'll be discussing cell organelles in next class. Thank you.